So why is research in MS important? Well, obviously, there's no cure for MS, as you've seen, and that's the goal for us, is to find a cure. I would like to warn everyone, I'm glad that I'm the first speaker this morning because everyone's had coffee and is slightly awake. But the warning is that neuroimmunology is extremely complicated and complex. And it's a twofold issue. Part of it is terminology. So a lot of the words that we use are just a foreign language to you. And if we explain to you what the concepts are, you will understand it. But the term we use, you really don't understand that. So there's terminology issue, but it's also that it's an extremely complex system, which is that the immune system and the nervous system alone is never one color. It's never black or white. It's all different colors, and then you have to mix it all together, and you always get brown. So you have to be able to understand that and think about that when you study these diseases. So I wanted to give you the little warning that at some point this is going to get a little science heavy. If you have any questions, please ask. If you're bored out of your mind and fall asleep, I won't point you out. It's okay. No. So our goal, obviously, is to cure your disease. So how do we get there? Well, one thing that we need to do is we need to be able to study you and your specimens. We've been working on animal models for a really, really long time now, and we're not where we need to be. This is difficult with MS because MS is a disease of the central nervous system. So we cannot, well, we could, but it wouldn't work very well if we came up to you and said, hey, do you mind giving us a slice of your brain so that we can study it? That doesn't always go over very well with living people. So it's much more difficult for us to study MS than it is to study, for example, rheumatoid arthritis, where you can say to someone, do you mind if I take a sample of your synovial fluid from your joint? They don't really want to do it, but it's not, a lot of people do that. So research in other autoimmune diseases has come a lot further than MS because we're able to study it in humans easier. So once we know more about the disease, we can use more targeted therapies to better treat you. And like I said, MS is a disease that's been around for a long time. So why aren't we there? So I'm going to give you a little bit of history of the disease. It was first described in 1868 by Charcot. 1935 was the first laboratory model of MS. It was a virus-based model, and it was the first time that the suggestion of MS being an autoimmune disease came about. In 1948 is when the multiple antibodies were discovered in the spinal fluid, and that's why for your initial diagnosis, many of you had a spinal tap, and that's to look for antibodies. That is part of the diagnostic test suggestive of MS, and that was brought about in 1948. Now, the interesting thing is that the major discoveries in the immune system didn't start until 1970. And between 1970 and 1990 is when we learned a lot more about the immune system, which is kind of just the cells that are involved and their basic functions. We've learned a lot more since 1990, but the pattern here is that we knew about MS way before we knew about immunology. So that obviously hindered us. In, 1993, interferon beta was the first approved drug. And interferon beta is actually an antiviral cytokine that your body produces when you get infected with a virus. And we use it, obviously, to treat MS, and it was the first approved therapy. So a long time ago, back in the day, MS was classically defined as a virally triggered autoimmune disease, and that's where interferon beta came from. All right. So I wanted to just put in perspective how long we've had laboratory models of MS until today. Okay, so on the lower right-hand corner, this is actually one of my favorites. In 1935, the jockey filed the first patent for underwear. Okay, that's how long we've had laboratory models of MS. Amelia Earhart flew from Hawaii to California. And this guy here is Charles Darrow, who actually invented Monopoly. And here he is showing Parker Brothers and Milton Bradley the game. Parker Brothers rejected the game, saying it had 52 fundamental errors, 2 of which being complexity of play and length of the game. And they were absolutely right. But this is how long we've had animal models of MS, and we're still not where we need to be. So what's the problem? Well, MS is a unique disease, and that sentence really doesn't mean anything. So I'm going to explain what it means. The reason why it's unique is that it doesn't have all the characteristics of a classical autoimmune disease such as rheumatoid arthritis or lupus. It is a little different. It's unique in the sense that it's difficult for us researchers to study because we cannot, pull your brain or spinal cord at any time that we want. So we have to use these laboratory models. And up until now, the laboratory models that we've been studying do not fully mimic human MS. So another reason why it's unique is that we've learned the most about what causes this disease or the immune components of this disease based on clinical trial and error. So we've learned which cells seem to be the important players based on what therapies actually work best in clinical trials and what therapies have failed. That is usually not the case for most diseases. So what we have learned is that MS is an immune-mediated disease, at least initially and for some period of time, and that B cells likely play an important role. So what we don't know is we don't know what causes MS. We don't know if the immune system directly attacks the nervous system or if the immune system directs the nervous system to attack itself. And we don't know what the B cells are doing. We just know that they're important. So how do we design better therapies for you? And some of these I just talked about in the last slide, but we have to answer what we don't know. We do not know what causes MS, and we are working on this with collaborators at other universities. People have been working on the cause of MS for a long time now. We may know in 5 years, we may know in 20 years, we may never know. We just don't know. So in order to find the cure, although we're working on the cause of MS, we have to think outside of the box because we don't know what causes it. So like I said before, we need to know about the immune system and its interactions with the central nervous system. We need to know what the B cells are doing. And some really important research that we do is trying to understand how current therapies or future therapies that are going to be approved work. And the reason why we need to understand this is what I just told you. We've learned the most about the disease from the clinical trials and errors, okay? Or successes and failures, I should say. So if a drug works in humans pretty well, we need to understand how it works. And surprisingly to all of you, the drugs that work really well, we don't really fully understand exactly how they work. We just know that they do work. We have some idea of what cells they play, but we don't know the intricate details of how they work. Okay, so I'm going to get a little sciencey on you guys. The immune system is extremely complex. We are not studying the heart, and that's it. There are multiple immune organs throughout the entire body. See, all of these are different immune organs in the body. You have immune cells in almost every tissue in your body, and the immune cells in each one of these organs, even though it's the same immune cell, they can all act different, and they can talk to each other in a different way. So it gets very complicated. To add to that complexity, there are many different cell types within the immune system. On the left are cell types that are involved in what we term innate immunity. That means these are your first line of defense cells. So when you first see a virus, these are the cells that respond first. On the right are your adaptive immune cells. These are the cells that provide immunologic memory so that you see a virus, and then later on, if you see that virus again, your body immediately attacks it and you don't get sick. That's from these cells here. Now, the complicated part of this is some of these cells here can have these functions. There's cells in between. So it's a very complex system. All right, let's add to the complexity a little bit more. Immune cells talk to each other but also to cells outside of the immune system. And I put this up here not because I'm going to go through every single thing, but the idea here is that one cell in the middle can secrete all these different things and act on all these different cells, and then this cell can talk to this cell, which secretes this, which can come back and talk to these cells, and it does it all within the immune system, but also with all cells outside of the immune system. So it gets very, very complicated in trying to understand exactly what's happening with the immune system at any given point. So we're going to talk about B cells today because we know that B cell directed therapies work pretty well in MS. This is an electron micrograph of a B cell. A B cell is actually a round ball, and all of these little processes sticking off are different things that are on the cell surface of the B cell. And all of these can have different functions and different signaling events and can talk to all these different cells. So B cells were first discovered in birds. A common misconception of B cells is that B stands for bone marrow because B cells are derived from bone marrow. But B actually stands for bursa of Fabricius, which is this little organ here in the bird. It's very close to the cloaca, and I don't know if any of you guys know what a cloaca is, but it's really gross. And the bursa is very close to that organ. That's how I always remember where it is. Okay, so T cells, which we're not talking about today, I'm just saying this. T cells are derived from the thymus. So T comes from thymus, but it's the B cells where people get confused and think that B comes from bone marrow, where it actually comes from bursa. Okay, so what do B cells do? As I said, we think B cells are important in your disease. Part of your diagnosis includes the presence of antibodies in your cerebrospinal fluid. So B cells have membrane receptors that we call B cell receptors. They have lots and lots of different receptors on their surface. This particular receptor, B cell receptor, binds antigens. Antigen is another ambiguous term. What does that mean, right? So antigens are anything that the B cell receptor can recognize and bind to. So an antigen can come from a virus, an antigen can come from bacteria, an antigen can come from yourself. And when it comes from yourself, that's obviously not always a good thing. So I'm going to use the term antigens, and that just means whatever the B cell receptor is binding to, okay? So when the B cell receptor is membrane bound on the B cell, we call it the B cell receptor. When the B cells get activated with antigens, they go through this differentiation process, and they end up secreting their B cell receptor. Secreted B cell receptor is called antibody, okay? So these antibodies float around in your serum, and they bind antigens. So secreted antibodies can activate B cells and other immune cells to fight off pathogens, which is obviously a very good thing. We want that. If these antibodies are specific for self-antigens, that obviously can be very bad, and you can get all sorts of autoimmunity. Okay, so B cells do a lot more than just make antibodies. They can also serve as what I like to call the traffic signal of the immune system. They can tell other cells to stop or to go, or to yield even. They even have a yellow up there. So when a B cell binds to the antigen, or it actually can be activated in a lot of different ways, it talks to other immune cells through a variety of mechanisms. It has things on its surface, like I showed you on the electron micrograph, that talk to other immune cells that also have things on their surface, and those are called receptors and ligands. It can internalize this antigen into the cell and process it, and then it presents it on a molecule called MHC class II that other cells respond to. So this is usually a go signal, okay? This can be a stop, go, yield. This is a go. And B cells also can secrete all these soluble factors. And I showed you that picture of when I said the immune cells all talk to each other, they secrete all these things. Depending on the environment, those things can be a go, they can be a stop, they can be a yield. So B cells actually have a lot of functions outside of just making antibodies. So what do B cells have to do with the brain? Let me give you a very brief neuroscience background. So in your brain, as you all know, you have neurons. And oligodendrocytes are the cells in your brain that make the myelin that wrap around your axon, okay? So we have neurons, oligodendrocytes. Here we have microglia, which I like to refer to as the policeman. The microglia are always surveilling your CNS, looking for any sort of invading pathogens, and if they see it, they respond to it, they get activated, and they start talking to all the cells around it and say, hey, everyone, we need to get this guy in jail, okay? I guess sometimes they write warning tickets as well, in which case, not a lot is done at that point. They don't see that whatever is there is going to cause that much damage. So the astrocyte I like to refer to as the nurse of the CNS, and its job really is to support all of your major cells in the CNS. So we believe that B cells either directly or indirectly talk to cells in your central nervous system. And we believe that this is done through an orange telephone, going through possibly the policeman to the nurse, but we're not exactly sure. It's very complicated science. And this is an old school telephone. It's not even a cell phone. We don't think the B cell has the capability of using a cell phone yet. Okay, so how do we study this? So on the left are our B cells, okay? These are the round balls. When B cells are unactivated, their surface sort of looks like this. And when B cells become really activated, their surface looks like this. And they push out a bunch of things and all this stuff, okay? So these are astrocytes from the CNS. So what we do is we take primary cells, which means they're not a cell line, they come from humans or animals, however we need to get them. And we activate the B cells, and then we dump them on top of the astrocytes and see what happens. So I'm going to show you a little bit of data, and it gets obviously a lot more complicated than this, and I will explain this to you, okay? So GFAP is just a protein expressed on astrocytes that when astrocytes become activated, they upregulate expression of GFAP. So this measure on the y-axis is cell size. So what we do is we activate the B cells, we dump them on the astrocytes, and then we look to see what the astrocytes are doing, and we run it through this very fancy machine that looks at these cells on a per cell basis. So in unactivated cells, you have a range of size of cells with, this is actually a density plot. So you have a little higher density at the higher size, a little higher density at the lower size. But if I make this gate here and I say how many of these cells are actually GFAP high, there's not so many. Now if I activate the astrocytes, what I can see is that we have a larger density of cells that are increased in cell size, as well as we have more cells that are GFAP high. So we're in the beginning stages of this. This is actually a novel technique that we developed in the lab because the way that these experiments are classically done is looking through a microscope and taking pictures of these cells under the microscope, which we started doing. And the problem that we had was that it was very difficult to actually quantitate what's going on. So we developed this technique so that we could better quantitate. So I can actually get percentages. I can actually measure the difference in cell size rather than just taking a picture and saying, hey, look at these two pictures. Don't these cells look bigger? I actually have numbers, so it's more believable. So we found some interesting things with the B cells and what they can do to astrocytes. Sometimes they activate them, sometimes they kill them. It depends on the stimulus. So as I said before, we also study mechanism of action of drugs, and we really like to study Copaxone, which some of you are probably on. It's also called glatiramer acetate. And we like it because it is the safest therapy that they have for MS. It does work really well in a certain percentage of patients. So if we can understand exactly how it works, we may be able to combine it with another therapy to promote its efficacy. We may be able to talk to Teva about reformulating it in some way to make it better. We don't know exactly where it's going to lead us, but our goal is to figure out how these drugs work to make them better. So this is another experiment. So what we do in the lab is we take the drug and we conjugate it to a dye. The dye is called 488. That means nothing. What that means is that the dye is excited at 488 nanometers. That's all that means, okay? So we have 488 alone, and this is a B cell dye that we use. So these cells are B cell negative, these cells are B cell positive, and if we use the 488 alone, we don't see any positive cells, either B cell negative or B cell positive. If we conjugate Copaxone to 488, then what we see is there are some non-B cells that bind to Copaxone, as shown here, and there are B cells as well that bind to Copaxone. And we've taken this a lot further, and where we are now with this is we actually understand down to the specific molecule on the B cells and on these other cells what Copaxone is binding to. So we know what it's binding to, and we have a little bit idea of how that changes the phenotype of the B cells. So we've done pretty well there. Okay, so how do we prevent disease? This says, no, not that one, the big one on the top shelf. So everyone's favorite needles, vaccines. So most of us think of vaccines in the sense of being immunized for pathogens so that when we encounter it again, our immune system immediately attacks it and we don't get sick. But the vaccine strategy in MS is much more complex because we don't know what causes disease, okay? So, lots of terminology here. I will explain everything to you. These are just words, but the concepts I think you will understand. So, what do we know about MS? So, we believe that designing a viable vaccine strategy for MS involves exploiting the known mechanisms driving this disease, and there are 4 of them. So I'm going to tell you exactly what they mean. This is an extremely complicated slide. I really don't expect you to understand all the cell types involved, but it's important to just understand the concept of what the words mean, okay? So I'm going to use the same example over and over and over again to describe all of these so that you understand this. Okay, molecular mimicry. When I explain this to you, you'll say, oh yeah, that totally makes sense why it's called molecular mimicry. So molecular mimicry means that when you are infected with a virus, I'm using this as an example, okay? When you're infected with a virus, your immune system somehow sees that virus and gets confused and thinks that portions of the virus look very similar to, mimic, portions of your central nervous system. And I'm going to use the neuron as an example, okay? It may not exactly be the neuron, but I need to give you guys some examples. So molecular mimicry means that you are infected with some sort of pathogen, we're going to use a virus, and your immune system gets confused, it goes to attack the virus, and then it thinks, oh, my neuron is the virus, okay? So the virus is mimicking your neuron, okay? So it's molecular mimicry. That makes sense, right? Okay, so then we have epitope spreading. And what that means is that when you get infected with, let's just say, a virus, and it infects cells of your central nervous system, when you get infected with the virus, what your body does is it tries to eat the infected cells, or the infected cells die and they just dump all their contents. So you have cells in the central nervous system that are either infected or other cells are infected. It's causing this reaction, and you have the dumping of neuron components, let's just say. So your immune cells are there to fight the virus. They pick up parts of your neuron because they're there. And initially, the response is to a very small portion of your neuron, and the immune system fights that, and usually everything is cleared and nothing happens, okay? Now, sometimes what happens is that your immune system fights that little part of the neuron, but it doesn't get cleared. So you see this a lot with a chronic infection. And because it's not getting cleared over time, you have what was a benign response to a little part of your neuron. Your immune system attacks more and more and more and more of your neuron to the point where it now becomes a problem. And now you're getting MS-like symptoms. So this is called epitope spreading. The epitope part being, let's say this is your neuron, the benign response started over here, you spread on the epitope of your neuron, okay? And then you have a problem. All right, so cryptic antigens, this again is just terminology. It basically means that all of us, people with MS and not with MS, have autoreactive cells all the time. We are always presenting self-antigens. Most of the time, we don't respond to self-antigens. There are some cases in which something is happening. Let's again use a viral infection as our example, that secretes certain cytokines or whatever and provides this environment where these normal antigens, self-antigens that are constantly being presented are now presented in a different way, okay? So when these antigens, these self-antigens get presented in a different way, your body sees it as foreign, and then you end up attacking yourself, okay? And then you have bystander activation. So I already told you we all have autoreactive cells all the time. They always bind self-antigens. We're always presenting self-antigens. Most of the time, it's not a problem. In some cases, so we'll use MS as an example, you constantly have immune cells coming in and out of your central nervous system. And some of them bind self-antigens, some of them bind myelin components, whatever they bind, okay? If something is going on in your central nervous system, let's say you have a virus infection that has nothing to do with the autoreactive cells, okay? You have a virus infection, it's in your brain, you may not even know you have it. It could be silent, your body is fighting it, no issue. What happens though is that as these autoreactive cells are coming in and out of your brain that are binding self-antigen, that are normally quiescent, these cells that are responding to this virus in your brain, and again, you may never even know that you have this infection, activate these autoreactive cells in what we call bystander activation. So they don't mean to activate it, it's just your autoreactive cells are kind of innocent bystanders of something else that's going on. So we think that all of these things happen in MS, and although we don't know what causes MS, we know that these mechanisms seem to be happening in MS. So how do we exploit how do we exploit this to develop a vaccine? Well, for molecular mimicry, epitope spreading, and cryptic antigens, we actually have to know what the antigen is in MS to design a viable vaccine strategy. We don't know what the antigens are, okay? So in bystander activation, we don't need to know the antigen for this. What we need to do is be able to program immune cells to talk to the autoreactive cells in the central nervous system and tell them, hey, calm down, okay? So remember I show you the B cells can be a green light or a red light. We need the B cells to act as a red light in the central nervous system, okay? So I should maybe give you an update on this. We have been working on this for some time now. In our initial studies in animals, the vaccine did not work at all. And when we pulled the cells out of animals, what we found is that the animals had no response to the antigens we were immunizing them for. So the vaccination and the boost actually didn't work in the animals. So where we are now is we've come back and we're actually looking at a panel of different CNS antigens that immune cells actually respond to. And it's very surprising that there are immune cells that respond to absolutely, one CNS antigen no immune cells respond to. Another CNS antigen some respond to. The issue, and I didn't talk to you about these other cells, is that what we find is that if immune cells are responding to the CNS antigens, a lot of different immune cells are responding to them, not just one cell. Some of the cells that are responding to those CNS antigens are actually killer cells. They're CD8s, they're NK cells. And what happens when they're activated is they come back and they kill the other cells that we want to promote the immune response. So what we're having to do is get rid of all the killer cells that are responding and just promoting the good cells that are responding. So it got a little more complex than what we had initially anticipated, but it's moving. This is a very complicated thing. So how do we cure MS? So the only way to get a cure is either a twofold process, which is stop your immune system from attacking the central nervous system. And there are some relatively decent therapies out there that do this, although we don't fully understand exactly how they work. And at the same time, we have to repair the damage that's already been done. And we work on this too, mostly in collaboration with other labs. The other way to get a cure is to develop a vaccine. And as I explained to you, the vaccine strategy for MS is very complicated, and that is because we don't know what causes it. So we're trying to get around that and manipulate the immune system or reprogram the immune system in a way that gets around the fact that we don't know what causes MS. So I say this, we must work on this together and help us help you. And the reason why I say that is because if your neurologist is at the Rocky Mountain MS Center, there may be a time when you come in where they ask you if you would be willing to give an extra vial of blood. They will have you sign a consent. And the reason for that is because they are all blood-sucking vampires. That's it, no. No, the reason for that is for us, and it's for us in the research lab. We need to study your blood cells. We need to know what your B cells do to the astrocytes. We need to know what your immune cells respond to as far as CNS antigens go for the vaccine. So if you don't mind donating, during a standard of care visit, you're already giving blood. So it would just be an extra tube. And if you don't mind, this is what it's for. So that's why I say help us help you. Okay? Thank you guys. Thanks to the Rocky Mountain MS Center, Karen and her staff, Dr. Vollmer and all of the doctors. Thank you guys. Have a good day.